And with that, we are done with our state and U.S. updates, and I will welcome our colleagues to the north. I see Chandra coming on. Are you going to kind of be emceeing this for
I think we
All
can go with
right,
that
cool.
plan if you
I
wanna
will kick it
fade
over to
into the
you and
background there.
let you run the show for a little while here.
Okay, amazing. Thank you so much for having us. My name is Chandra Moffat. I'm a research entomologist with Agriculture and Agri-Food Canada on the West Coast in British Columbia. So let's see if I can share my screen. Makes me a little nervous after the last session. I'm just going into slideshow mode now. What do we see? I'm not seeing slideshow mode. Lauren, you want to help me out?
That's good.
Oh,
Yeah,
there you go. Yeah. Looks
Looks
like you
good?
got it.
Okay,
yeah.
amazing. So we've had Canadian presenters at the summit before, but it's new to have a Canadian session. So we'll have three talks today. The first will be myself, as well as Lauren Marteaux, and we are co-leads of a relatively new project funded by our agency, Agriculture and Agri-Food Canada. And today, our three presentations will touch on Canadian research efforts for spotted lanternfly management and risk assessment. So Lauren and I are going to kind of swap back and forth here through our presentation. Hopefully, it's fairly smooth.
Yeah, we'll see. Okay, so unfortunately, as we all really predicted, spotted lanternfly sightings are on the rise in Canada. This is primarily in the eastern half of the country, and a particular concern is Ontario and Quebec. So we see these are sightings generally dead or alive. We don't have a full infestation yet, but these are on the rise, and we are seeing a lot more live sightings. And this is a big concern for our nursery and fruit and wine industries. Next slide, Chandra. Okay, so if we take, for example, Southern Ontario, this is an area with very high density of vineyards. And if we hit next one more time, this is where the lanternfly sightings have been. So, bit of a concern. Go next. This is really no surprise, though, considering how far north this pest has been making its way in the United States. It's now around Detroit, so we really are surrounded on the shores of the lake and now to the west as well. And so that brought about this project in response from federal government from Agriculture and Agri-Food Canada that Chandra will introduce now.
Yeah, so our project is it's building on efforts by other federal agencies. So Canadian Food Inspection Agency is our regulator for all plant health threats. So they have a surveillance program that we'll speak to in a moment, as well as our colleagues with Canadian Forest Service will be presenting later. But in speaking to our senior management about the risk of spotted lanternfly to Canadian agriculture, they invited us to submit a proposal almost 2 years ago. And so we assembled this national team focusing on researchers primarily sort of in that Ontario, Quebec region, as well as in British Columbia, but with colleagues all across the country. And so it's been a large project, and we've just applied for an extension. So everyone cross your fingers for us that our project will continue to be funded. In the first phase, so we have four research objectives. And so while Canadian Food Inspection Agency has surveillance efforts across the country, it's focused primarily on transportation corridors and other trapping networks that they have. So ours was to create a monitoring network specifically for agro-ecosystems to promote early detection amongst producers, and to really try to, catch any spotted lanternfly that end up anywhere off of those major transportation routes where the current surveillance efforts had been. Developing new spotted lanternfly attraction tools, which Lauren will speak to, biological control for Tree of Heaven, which is something I've been working on since 2019, and development of biological control for spotted lanternfly with entomopathogens. Back to you, Lauren.
All right, so yeah, as Chandra mentioned, Canadian Food Inspection Agency has been monitoring for lanternfly across the country. So these dots here on the map, and they are focusing on the transportation corridors and anywhere that a lanternfly has been sighted. But we want to, of course, focus on farms. Next slide. So we put together this surveillance team across the country, and in 2025, we did some scouting at 32 sites across 6 provinces. Next. So we focused on vineyards, but also farms, some of our research stations, and public parks. And this was primarily visual scouting of, for example, the vineyard perimeters, posts, trees in the area, and in some sites also some trapping. And happy to say that we did not detect any lanternfly in these sites this year. Next. But so I work in Harrow, which is a tiny hamlet in this blue triangle here, about an hour south of Detroit. And of course, I'm really concerned about any sightings we find because I'm really surrounded here. And I want to make sure that we're responding as quickly as possible. And when it comes to this biovigilance this early in an infestation, the public, it's really a critical piece of surveying. So next. I put together a project on iNaturalist, which allows me to track all the observations of tree of heaven and spotted lanternfly in Canada, and it arranges them by the most recent observations. So I can check this every few days just to make sure I'm up to date. Next. So October 3rd rolls around this year. I'm having a leisurely morning on my way to a conference, and I see that someone observed a live lanternfly 15 minutes away from me at a conservation area. So I ran over there, over to this tower where the observation was. This is a favorite perch of birders, as you can clearly see from this picture. But so when I ran up there was a family of birders up there. And after catching my breath from those stairs, I asked them if they happened to have seen a lanternfly. And it turns out that they were the ones that did make this observation on iNaturalist. So I thought, oh, great, where is it? They said, oh, well, we let it go in the bushes. Why? Is that a problem? So unfortunately, we could not find it. I retraced their steps throughout this conservation area. I logged all the tree of heaven, all the black walnut. I've been there twice scouting and could not find any lanternflies or egg masses, although I will go back later this month. But basically, next slide. This led me to try and increase our public engagement so that people are more aware of this pest. So I got Canadian Food Inspection Agency to send some signage for us to put up in public areas. We've been doing some social media posting, and I also coordinated with the local conservation authority to try and put up posters in the area and train their staff on scouting for this pest. Okay, next. So moving on to the attraction tools for this. You probably know that we don't have a very effective lure for lanternfly. The primary approach is to look at volatile lures, but because this pest also communicates through some acoustic drumming, that could be an avenue to explore as well. Next. But one does not simply dive into this without first consulting Miriam Cooperband at the USDA, because she's been working on lanternfly attraction for a decade. Now she has tested chemicals from all these stages of lanternfly squished versus intact, honeydew, tree of heaven, you name it, and nothing has really outperformed methyl salicylate, which is a pretty ubiquitous chemical released by stressed plants, and it's not at all targeted for lanternfly. So we do have plans to work on some of these vibroacoustics with her team in coming years. But one of the things that hadn't been tested was the waxy coating on the egg masses. And we know that lanternfly tend to lay their eggs in clusters, multiple clusters on the same area of the tree. So is that because the adults are attracting each other, or is it something about the hot trees? Well, maybe it is something to do with the wax. So next and next. So in November 2024, I went over to Toledo, and big shout out to Amy Stone and Jonathan Shields because they let me interview them and took me around to show me where the infested sites were so I could sample some of this wax. So I scraped wax off a number of the freshest egg masses I could find and also sampled bark next to it. I don't know how many people have dealt with this wax, but when you go to scrape it, I mean, it looks substantial, but as soon as you scrape it, it basically fluffs off into the air like ash. It's very fine material. It's very strange. Anyway, after some struggling, I did get about 5 or 6 wax samples, and I brought those to our chemist in London, Ontario, who did some fatty acid profiling via GCMS. Next. Okay, and so basically what we're seeing on the top graph is the fatty acid profile from the tree bark, which is essentially the blank, and below is what we see on the waxy coating. So there are a number of different peaks there, fatty acids. Next. Now, I'm not a chemist. I know nothing about this, but Justin tells me that generally insect waxes are dominated by longer chain fatty acids, whereas the peaks we're seeing unique to the waxy coating are much shorter. He's wondering whether there's some residual egg in these samples. So we now need to go and just test pure eggs compared to the waxy coating. And we now have access to some fresh egg masses because we have a colony in London, and our GCMS, which was broken for most of last year, is now repaired. So we'll be looking into that soon. And then basically on to you, Chandra, for the other portion.
Okay, great. Thanks, Lauren. So as we know, and we've heard reference to today, spotted lanternfly has a close association with tree of heaven, not as the only host tree, but as an important tree in the spread of spotted lanternfly. So for those who aren't aware, tree of heaven is a deciduous tree that's native to China and other parts of Asia. It's dioecious, so it has male or female flowers. And these compound leaves, but with smooth margins, it can be confused for things like sumac. But it's different. It turns into a large tree, not just sort of a shrub, and it has these maple seed samaras that proliferate, and it's a highly clonal species, so multiple modes of reproduction. And it's super invasive. Where it gets in, it just can completely go wild. So disturbed areas, like for most invasive plants, rail lines are a major corridor of spread, highways, so transportation corridors, exactly where we're looking for spotted lanternfly. Very invasive in agricultural lands, particularly vineyards, fields, hedgerows around agricultural matrix, and oak savannas and grasslands. So the global distribution of tree of heaven, it's highly invasive in North America and Europe, and has been popping up in South America, South Africa, Australia, and New Zealand. And the maps I'll show today are from 2025, as my technician, Tyler Nelson, has been on parental leave and hasn't been able to update them for me. So not too much has changed in terms of the real distribution, but it's great to hear there's more and more records that are being put in. So we take records from a variety of different databases, but as of last year, here's the recorded distribution of tree of heaven in North America in pink. And then again, these are 2025 data, but overlaying spotted lanternfly observations, and my map might not be quite perfect there, but we can see that the distribution of spotted lanternfly highly overlaps with tree of heaven. And when we look to the rest of North America, other parts of Eastern North America and Western North America, like California, Oregon, Washington, and British Columbia, up above the border where I'm located, there's extensive populations of tree of heaven that are just lying in wait for spotted lanternfly to make its way into those regions. Focusing in on the area that Lauren was referencing and where we have spotted lanternfly in close proximity to the Canadian border, I'll just zoom in here. And so we can see sort of in the beige peninsula there that dips down the area that Lauren was showing us, and that we have a lot of tree of heaven. And so again, tree of heaven lying in wait for spotted lanternfly to establish. So as I mentioned, I'm in the West in British Columbia, and so where we have tree of heaven here, it perfectly overlaps with our wine grape producing region. And so while we don't have the acreage of California or Oregon, we have about 2,400 hectares of wine grape production and more than 200 wineries. And literally, if you're standing in a winery in our region, you will see tree of heaven, in the winery on the road right beside. And so it's really set up as a perfect storm. So I've been working, our stakeholders here have been supporting me to work on biocontrol for tree of heaven since 2019. But as I became more aware of the risk that spotted lanternfly poses, it gets us to thinking, can we indirectly manage spotted lanternfly by controlling tree of heaven? And of course, we're hearing lots of folks mention, mechanically and chemically removing tree of heaven, but can biological control play a role? So last year in the summit, we heard from Scott Salem, who talked about the promising trunk-boring weevil Eucalyptus brantii, which has been petitioned by TAG and is awaiting approval for release. But there's another biocontrol agent we've also been working on, and this is a Eriophid mite, Aculus tahangensis, that's native to China, and actually has been also showing up in different areas where tree of heaven invades. So where tree of heaven is highly invasive in Europe, the mite has followed, and there's extensive adventive populations of the mite in Europe. As well, the mite has been picked up in several locations adventively in the US. But we're looking at it from a classical biocontrol perspective. So as mentioned, it's native to Asia, to China, Korea, and Japan. And so Eriophid mites have been quite successful in some programs as biocontrol agents, as they're highly host-specific, have a very high reproductive rate, high dispersibility, and they can have a high impact, especially on younger plants. So this tree of heaven seedling here is one of the photos from our collaborative work in Italy. This is led by BBCA, the Biocontrol and Biotechnology Institute of Rome, Italy. where they've been working on the information that we would require to support a petition for release of the mite first into Canada, but then with our US collaborators, there could be a follow-up petition for release into the US as well. So I'm just going to show you here a summary of some of the work that we do. And so this is led by Francesca Marini in Italy. And so when we conduct biocontrol programs for invasive weeds, we use a phylogenetic method of selecting potential non-target hosts. So working from the most closely related out to the most distantly related. So at the top are the most closely related. So in North America, there's three native species in the same family, Cimrambaceae, as tree of heaven. Castella amori, Lentinaria floridana, and Lentinaria pilosa. And there's always some taxonomic considerations here. So we're aware of that if in case there's any experts in the audience. But those three species are native to southern areas of the US. And so those are a very high priority to test. Even from a Canadian perspective, when we release biocontrol agents for invasive weeds, we are very united, Canada, the US, and Mexico, in reviewing each other's petitions and making sure that any agents released don't pose an ecological threat to native species anywhere in North America. So Francesca's been working diligently to look at if the mite is able to subsist on any of the species that we selected to be tested. And in what we call the physiological host range testing, which is very experimental, small scale, mites were able to survive and develop on 2 of the native species at very low levels. So what this looks like is when we look when plants are set out in the field with the advent of mites, the number of mites that can develop. So on Tree of Heaven on the left, a very high number of mites. We're looking at thousands of mites, 12,000 mites per gram of plant biomass, whereas on those native North American species, highly, highly reduced, like order of magnitude less, like less than 200, or on Lentinaria, less than 20 mites per gram of plant tissue. So there is a small ability for the mite to feed on these plants. However, over time, the mites are not able to stay alive and they do die on the plant and they're not able to continue and have a viable population going forward. So the risk is considered quite low. Looking at the ecological host range, when we took this out more into the field environment, the mite was not able to sustain on the 2 native species. So of all the native species to North America that we tested when we did them in the field, the mite was not able to cause damage or reproduce and sustain on any of these plants. So the mite is very host specific. And so we're currently working, my lab, our collaborators with CABI in Switzerland and BBCA on a joint petition for release of this mite species into Canada. And the 2 different biocontrol agents that we're looking at here, they target 2 different stages of Tree of Heaven. So whereas the trunk boring weevil has been shown to be potentially a very highly efficacious agent for large trees, the mite goes after seedlings, so could play a really good role in reducing the spread of Tree of Heaven clonally. We've been looking as well at the ability of the mite to colonize different genotypes of Tree of Heaven. And so while there's not overall a statistical difference here, we do observe some differences in the mite's ability to colonize and damage Tree of Heaven seedlings from different parts of Canada, east versus west. And so when we look across, the distribution of Tree of Heaven, we have invasive genotypes of Tree of Heaven all over North America and Europe. And we don't necessarily know the genetic relatedness of those genotypes and how they may be susceptible differentially to biocontrol. So we've initiated a genomic sequencing project for Tree of Heaven. It's a global project, and so far we have samples from Canada, Switzerland, the UK, Italy, and New Zealand, but we're looking for more samples. So I have a little link here, and you're welcome to contact me as well. But anyone's welcome to contribute Tree of Heaven samples to our genomic sequencing project, as well as because the mite is inventive in the US. We have in our renewal we've asked to do a complementary sequencing project on the mite, the genomics of the mite, and its inventive populations in Europe, North America, and native populations from Asia. So if you work in an area with Tree of Heaven and you know you have the mite, we would also be very happy to accept mite samples and collaborate with you. And so that's the end of this presentation. Again, there's the link to that, and you can email me for more info. Thank you. We're now going to transition to Michelle Franklin, who will present the next section on entomopathogens.
Can you hear me okay? Okay, I'll just get my screen shared. Can you see my screen all right?
Looks great, Michelle.
It looks good? Okay, perfect. Okay, so thanks for inviting us to give an update from Canada. I'm super excited to be able to share some research that I've been doing in collaboration with Tara Garepi from our London Research and Development Centre. And this work is looking at entomopathogen tree band for biological control of spotted lanternfly. So our goal here was really to develop a targeted delivery system for entomopathogens to kill spotted lanternfly. The rationale here is that we could exploit spotted lanternfly's climbing behavior as well as this crowding aggregation behavior with a targeted entomopathogen delivery system. And so our approach here is to use a burlap band, and this could be wrapped around an infested host tree, or it could be used around a fence post, say in a vineyard situation. And ideally, in the future, we'd like to combine this with an attractant. But as we've talked about, we haven't quite found the right pheromone or vibroacoustics yet to develop this kind of attract and kill based system. So we wanted to build on some research that Dr. Ike Shikano and Kristen Street have done from the University of Hawaii. Dr. Shikano has led the development of an oil-based carrier, and this oil-based carrier is able to deliver a commercial entomopathogen. Their use was for taffritid fruit flies when they made bait stations for the taffritid flies to contact this entomopathogen. We've looked at repurposing this, and we have an agreement between Agriculture and Agri-Food Canada and the University of Hawaii to test Dr. Shikano's oil-based carrier formulation in this tree band approach. So a couple of questions that I'd like to address in today's talk. This is kind of the beginning stages of this research. It's our first year. And so first question is, do these entomopathogen tree band formulations containing commercial Beauveria bassiana strains cause mortality in spotted lanternfly? And then the second question that we wanted to look at is, if we're to use these outdoors, how long do the Beauveria spores remain viable on tree bands when we put them outdoors under field conditions? So this is a look at our setup. We received this oil-based formulation from Dr. Shikano. We purchased commercial Botanigard, which is the Beauveria bassiana GHA strain. We mixed the Botanigard with the oil-based formulation. We then applied that to these burlap bands. And then this was used for the two different trials. The first was looking at the efficacy. So doing this in a laboratory setting because we are in an area where we don't have spotted lanternfly, and this needed to be conducted in PPC level 2A quarantine facility. This part of the work was really led by Dr. Tara Garepi because she has the quarantine facility at the London Research and Development Centre. And then we also used these bands in our field trials looking at spore viability, and that was work that was done both in London, Ontario, as well as in Agassiz, British Columbia. Looking at the laboratory trial, we had 3 different treatments. We used a container. We put the burlap band inside of that container. The first was just burlap on its own. The second treatment was burlap plus the formulant. And then the third treatment was the burlap formulant and our commercial Botanigard treatment. So we took spotted lanternfly adults. These were about approximately 90 days post-hatch. We put 5 adults into containers with each of the 3 corresponding treatments, and we left them there for an hour. We were able to expose in 2 of our replicate trials, 18 to 20 spotted lanternfly per treatment in each of the trials. After the hour, these spotted lanternfly adults were moved to cages with Tree of Heaven with the corresponding treatment that they belonged to. We monitored these spotted lanternfly for 14 days post-infection. To confirm if there was mortality caused by the Bovaria treatments, we collected the spotted lanternfly that had died. We then surface sterilized the spotted lanternfly with a bleach solution and then placed them in a humidity chamber and checked them daily for sporulation. And so the reason we did this was because we wanted to confirm that the mortality that we were seeing was actually caused by the Bovaria treatment. And so you can see here, we had many nice adults with lots of white spores of Bovaria, showing that they were definitely infected by the Bovaria treatment. Here's a look at our results. These are pretty preliminary since we've just completed these first 2 trials. You can see that in the burlap treatment and then the burlap plus formulant treatment, we had really low levels of mortality. This would have been just some handling mortality that we had in the first replicate trial, and then we had no mortality in the second replicate of the trial in that burlap and burlap formulant treatments. We are getting high levels of mortality when we have the Botanigard added to the formulant, which is good news. We're seeing upwards of over 70% mortality. And the gray shaded part of the bar shows you that we've been able to confirm most of these deaths due to Bovaria infection. If we look at the Bovaria infections over time, we see that we kind of have a peak in the number of dead spotted lanternfly around day 8, and 9 of this after exposure to the formulant and Bovaria. And so we have this kind of range between 5 to 11 days is where most of the mortality is happening. And this is pretty typical of a Bovaria infection, seeing mortality around this time period. So everything looks like it's on track in terms of that the formulant containing Bovaria is working to cause mortality using the tree band approach. So our next steps, we want to do a 3rd replicate of this trial with the adults to make sure that we're seeing consistent results. We also have done some preliminary trials with nymphs. We've done some with 2nd and 4th instar nymphs when we were developing the protocol, but we'd like to go back and revisit doing more replicated trials with nymphal instars. After this, we'd like to move on to larger cage type trials where we could wrap that burlap band around Tree of Heaven within a cage and look at exposure that way. And also we'd like to look at if we do have any passive transfer in cages in this kind of larger setting. Which would be great. We're also interested in trying another entomopathogenic commercial product. That's Lalaman's Lalagard, which is an M52 or MET52 product, Metarhizium bruneum. And we'd use the same methods, but just try a different entomopathogen and see what the efficacy looks like with that one. So I want to move on now to talk about our outdoor field trials looking at UV degradation of Bovaria. So we know that sunlight is a major factor that limits the spore viability of Bovaria. It often is rapidly degraded and has a short half-life when exposed to direct sun. This graph is taken from Jaronski 2010, and it's a graph showing the viability of spores on melon leaves in California, and the upper and lower surface of that leaf. And you can see from this graph that the lower surface of the leaf has a much slower reduction in the viability of spores compared to the upper surface of the leaf, kind of speaking to that UV exposure issue. So with our tree bands, we wanted to look at what the viability of spores would be in both the shade and direct sun outdoors. And so we did these trials both at the London Research Center as well as the Agassiz Research Center. I'm going to share the data today from our results from our Agassiz Research Center. So we had replicated tree bands on posts out at our center, and these were used to look at persistence. This is a better look at the setup. So you can see we had the burlap band, and then this had a UV shade that was around it, which is a repurposed dog collar that's used when we don't want our dogs to. So these worked quite well to shade the burlap, but we did find after some preliminary trials that even with just that UV shade, it wasn't really enough. And so we ended up coating this with a UV protective paint. And then we used a swab to sample the formulant weekly to determine the spore viability in the field. These samples were brought back to the lab, they were plated on media, and then we were able to look at germination of spores and spores that were not germinated. And so you can see in the picture there, those spores that are circled in purple are those that are germinated, and then you can see a spore that was ungerminated and circled in green there. So this is the type of graph I'll show you throughout looking at this persistence data. On the y-axis is the proportion of germinated spores, and then on the x-axis is the time after treatment. And the red line shows you that what the percentage of viable spores was in the formulant that was just mixed. And so we sampled this 3 times over the course of the study, and you can see that we had about 80% of viable spores throughout. When we look at spores that were on those burlap bands in the direct sun, we see that we have good viability of spores up to a week after being out in the field. So that's pretty decent if we think about, trap changes and stuff. A weekly trap change would fit into most IPM kind of programs. After 2 weeks, we did see no germinated spores in our plates in the lab. And so it was around a week that we had good viability of spores. When we look at the shade samples, the viability of spores remained pretty good for a long time. We saw upwards of 60% viable spores, even 3 weeks after we had put those burlap bands out in the field. So we're quite impressed with this result. This looks like we would have good viability, a good trap for a long time out in our fields. So a few take-home messages from the work that we've been doing on these tree band traps. First, the laboratory trials show that the Bavaria formulated tree bands work really well to cause mortality, and we have good levels of mortality in those initial lab trials with adults. Most of the death is occurring between 5 to 10 days post-exposure, and this is pretty typical of Bavaria entomopathogen. Outdoor exposure trials show pretty promising results too, with spores remaining at a good viable level, 1 week in direct sunlight and 3 weeks in the shade. And with that, I just want to thank everybody that's been involved. There was a lot of student help for the first year of this project. And then a special thanks to E.K. Chicano and Kristen Street for their help and advice, as well as the formulant. And our funding was provided by Agriculture and Agri-Food Canada. And we hope to continue to extend this work in a second project. So I'll turn it back to Chandra.
Thank you so much, Michelle. And next we'll have Amanda Rowe and Anna Turbelin from the Canadian Forest Service. Hi, Anna.
Hi, let me just load the slides. Okay, am I in presenter view?
It looks great. Good work.
Okay, perfect. So good afternoon. I am a research scientist at the Natural Resources Canada, and I will be updating you on work that I have been doing with Amanda Rowe, also a research scientist at NRCan, on the cold tolerance of spotted lanternfly. So basically, we've been trying to work out whether Canadian winter would kill spotted lanternfly or not.
Is it not advancing for you?
No, there we go, there we go.
Oh, there we go.
Yeah, thanks. First, I would like to thank our funding agency, so the Canadian Food Inspection Agency, but also Surge International, and all our collaborators and technical support that have been great help for both the projects that we've been running. I want to mention the Invasive Species Center, for the lovely little icons that you can see here. And just a small disclaimer that I did use some AI images, but I'm sure you'll be able to spot them within the presentation. Okay, so first, a quick overview of what we already know on spotted lanternfly cold tolerance, specifically the egg masses. This is based on work that we have previously done and published. We looked at egg mass cold tolerance strategy, and we found that SLF were chill susceptible, so they die from chill injury before they freeze. We also ran lower thermal limits experiments for short exposure treatments and long exposure treatments. Our short exposures were 1 hour and 12 hours, and long exposure treatments were 10 days and 15 days, and across 5 different temperatures from 0 to -25. There we found that hatch rates were significantly impacted in the short term at or below -20, you can see here, and in the long term at or below -15. Now, I did say that SLF was chill susceptible, but we had a hatch in our lowest treatment of 1 hour exposure at -25. And so we found that they hatch actually quite close to their super cooling point, which is the point at which they freeze and is normally not so helpful for chill susceptible insects. But in this case it is. So we suggested that the lowest supercooling point measured, -27.7, to be a conservative approximation of the lower lethal temperature for SLF egg masses or eggs. From the study, there are a few points that I'd like you to bear in mind and that have inspired our next research. These were that you can see variation in supercooling points. The range is like by 10 degrees here, from -17.4 to -27.7, and that's only across for 5 egg masses, points that were measured. Then we did find that the populations in Pennsylvania could tolerate temperatures at least 5 degrees colder than the ones predicted in South Korea. So here we see a potential population difference. And another point, final point here, is that our experiments were conducted in a laboratory setting. And that means that we collected them in the field, they were exposed, they were field acclimatized, and they were exposed to fluctuating temperatures before we collected them, but then they were kept at a constant temperature in our chamber until we did the experiments, and then back at a constant temperature until we put them out to hatch. Now, obviously, this is not what they experiment in the field, and they're more likely to get fluctuating temperatures. And there is evidence that that can impact positively or negatively the cold tolerance of insects, which could apply to spotted lanternfly and leads us to our next experiments, where we wanted to measure cold tolerance in the field at 3 different points in different locations for different populations. With the help of the government of Chicago and government of Illinois and government of Tennessee, as well as the USDA, Melodikina in Connecticut, we were able to collect egg masses from this location, Chicago for the northern edge and Nashville for the southern edge. As you would expect, the winters there look really different. In the presentation, and for interest of time, I'm just going to focus on Chicago and Nashville, mostly because egg masses from Westport were exposed to an artificial regime, and also our fall collection had a slight hiccup and was caught in a snowstorm, so we were not able to use those egg masses. To give you a small idea of the differences in winter, this is just like a few points for the profile. Illinois has a lot more days where the maximum temperature is sub-zero. That's 33 versus 3 in Tennessee. -10 days where the minimum temperature is below -10. Illinois is 20, Tennessee has 8. Tennessee, however, has more freeze thaw events, so that's like 60 versus 51 within that winter period, which was from, well, it was 24-25 winter. And we also looked at the temperature differential between the minimum and the maximum for the daily temperature. And there, actually, there's quite a much bigger difference in daily temperature between the minimum and maximum in Tennessee versus Illinois. So what we did, we deployed those lovely lampshade traps that you have seen throughout the summit, and following the Lewis et al. Instructions. We also added some temperature loggers. So you can see here we had a probe inside and one outside the trap. So this is a picture from Chicago, that mother tree that Scott showed you before. We did a first collection late fall. The dates in Chicago and Nashville are not quite the same, but the season kind of matches. So we had late fall here. We also collected midwinter and in April. Well, early spring. Once we collected the egg masses, we sent them to our quarantine facilities in Sault Ste. Marie. And we did 3 different experiments this time. We looked at the supercooling point. From 12 to 14 egg masses per season per location, and about 16 eggs, and that was more than 1,600 eggs to which we measured the supercooling points. We also repeated our experiment of the lower lethal limit with a 1-hour exposure, but I won't be sharing you those results today. That'll be for another time. And we looked at the hatch rate of the remaining eggs that were from the egg masses that we used for the supercooling point experiment. Now, preliminary result, actually, just what we found. Here you can see Chicago in purple and Nashville in blue. The supercooling points across season and for the locations range from minus 5.1 to minus 29.7. The median was about minus 22.5, but like you can really see, most of them are below minus 20, and actually 90% are below minus 18.3 degrees C. I don't know what the conversion is in Fahrenheit, I'm sorry. The effect of location on supercooling point was season dependent, and there were not really many differences, at least when using the modeling approach that we used, between Illinois and Tennessee in late fall and midwinter. But it was significantly different in early spring, where supercooling points in Chicago were about 1.2 degrees cooler than those in Nashville. And then for the hatch, we see a slightly different pattern for hatch rate in the remaining eggs that were used. And here you can see late fall median hatch rate in Illinois was 38%, late fall median hatch rate in Tennessee was 16%. In early spring, that went up. So early spring median hatch rate was 59% in Tennessee, 88% in Illinois. So you can really see an increase throughout the season, and also hatch rate in Illinois was consistently higher, at least in the late fall and early spring, than the hatch rate in Tennessee. So these are some of the results that we have found. We're still working on the rest of the study, but a few takeaway points based on both studies that we found. Actually, we could observe some plasticity in cold tolerance and observe like deacclimation in terms of supercooling points, which is biologically really quite interesting. In terms of management, it is still like below minus 20. The majority of them are still below minus 20. So whether you will experience minus 20 in April, where you are, may not matter for whether the impacted species are from an ecological point. Now, we also saw that hatch rate was significantly impacted. Survival of, sorry. There was a significant difference in hatch rate between egg masses that had been collected in the fall and then exposed to a constant temperature before being set out to hatch versus egg masses that were collected in the spring and then were set out to hatch. So here you can really see an effect of the fluctuating temperatures on survival, and it appears to be beneficial. We also observed no complete mortality at minus 20. The lowest temperatures in Chicago reached minus 20, and they did just fine. And that makes us suppose that a cold could be beneficial to SLF survival. Yeah, based on the results, which is not necessarily good news for us in Canada. With this, if you have any questions, I want to, well, first, thank you for listening, but Amanda and I are in the chat, so we're happy to answer any questions you may have.
Thank you so much, Anna. Scott, I can turn it back over to you.
Well, thank you all. Appreciate that. A lot of cool information there. Yeah, it looks like just kind of keeping an eye on things in the Q&A. There were a handful of questions. I think you're working through them. Oh, great. I think a lot of them were kind of geared toward Michelle's presentation. And it looks like she's powering through those. But if you prefer Michelle to stop typing and answer them live, we do have roughly 25 minutes left to discuss. So if there's anything you'd rather talk about instead of type, or we can, I'll certainly give it over to you. I see it came off camera, so go ahead.
Oh, I was just going to say, sure, I can. I think there was one more kind of talking about the Bovaria infection and potential for impact on other insects.
Yeah.
Yeah, Bovaria is a bit of a more generalist type of biological control agent. So I guess there is some risk that it could impact other insects, but I think kind of if we think about this as like in the future being more of like a attract and kill type of system where we're once we have that other piece where we have a pheromone or a vibroacoustic, then we really will be, you know, working on attracting these spotted lanternfly to the specific tree band. I think we do use these types of Bovaria formulations as spray formulations in, you know, greenhouse field settings. So the tree band would have less impact than, say, a foliar spray of Bovaria in the field. So although there could be an impact, I think it would be much less than if we were to spray it, you know, in a vineyard or something like that.
Michelle, I'll just add to that too. Like we used sticky bands on Tree of Heaven for quite a while for our monitoring. And so I don't know how it compares to other regions, but we had really very low bycatch. So if we're able to develop it into, you know, sort of a band around Tree of Heaven, again, I don't know about other regions, but in our region, we've had very few other species, mostly just earwigs and some ants that were ever caught on the sticky bands. So.
That's good to know. The other thing I wanted to point out that came up in the chat, and I don't think that I had said it clearly enough in the presentation, was that these tree bands, the formulation is meant to be like a contact, but not that they get stuck to it and die. We actually would like them to like contact the tree band and then leave and potentially be able to cause secondary infection to other spotted lanternfly that aren't on the trap. So we don't see, it's not sticky like a the tree band monitoring kind of system where the insects stick and die. It's sticky enough that it gets on the insect, but they're able to leave the tree band. The formulant is not that sticky that they're actually stuck to it for good. Yeah.
Sorry, let me come off of mute there. There are a couple more questions or comments coming into the Q&A. As an organic vineyard in Pennsylvania, is there any recommendations we should be implementing for SLF? I don't know if this is necessarily aimed toward you all or maybe just the panel in general. If there's anybody that wants to weigh in on any recommendations for organic vineyards, feel free. Julie's back. All right.
I got a similar question yesterday. I don't know if this is the same person or not, but my suggestion would be in the Penn State Extension Spotted Lanternfly Vineyard Management materials, there's basically that's all encompassing. So that has some organic recommendations as well as, you know, other options. And also kind of takes an approach to looking at a landscape level. And so that would be my recommendation to check that out. I can put that link in the chat. And then the other thing would be like tomorrow, nobody is speaking specifically about like organic methods, but Laura Nixon and Michaela Centenari are going to specifically be talking about what impacts SLF feeding has on grapevines. And like in the case of Laura, some issue with threshold. So again, I think you might want to tune in for that.
There's also a request in the chat from Matt Helmus about some of the information that Chandra provided with regards to Tree of Heaven and some of that information. Are you guys able to see the chat?
I was just looking at the Q&A, so I wasn't looking at the chat. Oh, yeah, good idea. Okay, thanks. I missed that. I'll put that in right
Cool.
now. Good idea.
Nothing else coming in. Julie, I like what you did yesterday. Are there any of the panelists that have any questions for any of the presenters today that are still on, or vice versa?
Yeah, this is Joanna Fisher with CDFA. I had just a quick question about, pretty interesting research that was going on. I apologize, I don't remember the presenter, but this was for SLF and Bavaria Bassiana. I was just wondering for the outdoor studies looking at spore viability, where were those conducted? And kind of what was the temperature, humidity, or if you know.
Yeah. So we conducted them both at our London, Ontario Research and Development Center, and then in British Columbia at our Agassiz Research and Development Center. And the data that I showed today was from our BC location. And the temperatures during that time would have been kind of highs of I'm going to say this in Celsius, 25, 28 degrees Celsius during the day, sunny kind of summer conditions. So they were exposed kind of to the conditions that we would be looking to put those kind of traps out. This was our first year, and we'd like to put those traps out over a longer period of time and into the fall too. So we'll be doing some more follow-up studies. We were just kind of working out our methods and the kinks and protocols and stuff this year with the method and kind of trying the different trap designs. We did definitely find that we used a UV resistant paint that's used in like craft supplies from Michael's, and that seemed to work really well to help block the UV and provide that protection.
Thank you so much.